Welcome. Let's look at week 2 of embryonic development. This is also referred to as the week of twos. And during the course of this lecture, we'll see why it is also referred to as the week of twos. The events that occur from the 7th day of embryonic development after fertilization to the 14th day is an event of the 2nd week of development. We have done a lecture on the 1st week of embryonic development. You should check that up before you watch this video so as to keep yourself properly aligned with this lecture. So we know that during the 1st week of development, we have a group of cells that are called the blastomeres that mitotically divide. And after they divide, there's a form of segregation that they present, which is called the blastocyst. And this blastocyst is seen to contain outer alignment of cells that are referred to as the trophoblasts. And we have inner cell mass that is referred to as the embryoblast. This is the final configuration of what the blastocyst is, and this is what is implanted into the endometrial wall of the uterus in the 1st week of development. The major events that occur in the 2nd week of development involves the transformation of the trophoblastic cells. And this transformation leads to the accomplishment of 2 events. The first one is to allow the developing structure to be completely embedded within the wall of the uterus. And the second event that will be accomplished due to trophoblastic transformation is the setup or the establishment of the uteroplacental circulation, which is the circulation between the maternal blood and the developing structure. So these are the 2 aims of the transformation of the trophoblast. And the second event that occur is within the embryoblast, which is the major structure that forms the embryo, which includes the entire body structure. And the transformation that they undergo is for the accomplishment of these following events. And this includes the formation of the bi-laminar germ disc, the formation of amniotic cavity, and also the development of the primary yolk sac, which also further transform into the secondary yolk sac. This is what the transformation of the embryoblast is set to achieve. So we have development occurring in the different regions of the blastocyst that we have in the 1st week of development. So let's dive in to see what occurs within the trophoblast and also the embryoblast in the 2nd week of embryonic development. We said that the event that occur within the trophoblastic cell is to allow the developing structure to completely embed within the endometrium lining of the uterus and also to set up the uteroplacental circulation. So let's see how this goes. First, the trophoblastic cell, we first differentiate into 2 types of cells. We have the cytotrophoblast and we have the syncytotrophoblast. This is the cytotrophoblast, the inner layer of the trophoblastic cell, and this is the syncytotrophoblast, the outer layer of the trophoblastic cell. So let's further dive in to see what these 2 subregions transform into. So for the cytotrophoblast, we say that the cytotrophoblast is the inner layer of the trophoblastic cell. This is the area highlighted in black. And if you can view this collection of cells critically, you will see that their cell wall is well defined. It is still intact, and they continue to proliferate, begin to increase in number due to mitotic division. They begin to proliferate and drive into the trabeculi of the syncytotrophoblast. This layer in the outer region is the syncytotrophoblast. The trabeculi is the space between the 2 lacuna. These are the lacuna, and the space between them within the syncytotrophoblast is called the trabeculi. So they begin to proliferate by transforming into a finger-like projection that will form the primary villi. The primary villi forms the bulk of the placenta, and it helps to increase the surface area through which the product of the maternal blood will reach the developing embryo or the fetus, as the case may be. And that is why you see this finger-like extension. This is just to increase the surface area of whatever product that will be exchanged between the mother and also the developing structure. And this is the finger-like expression. Then let's look at the syncytotrophoblast. The syncytotrophoblast is the outer layer. And if you look at the syncytotrophoblast critically, you see that the cell wall are ruptured. As the cell proliferates, there tend to be loss of their cell wall. So what you see within the space of the syncytial trophoblast is nuclei. So these dots represent the nuclei, and you see the nuclei enclosed in an entire wall. So the cells are contained like a syncytium, kind of, and that is where the name is drafted from. So they are collected together with loss of their cell membrane. So this is what they are presented with, unlike the cytotrophoblast that is seen with their cell membrane still intact, and they, of course, continue to proliferate and create a finger-like expression. As the syncytial trophoblast proliferates, it burrows into the wall of the endometrium. It breaks the wall, and that pushes it into the endometrium lining of the uterus, and that is how it begins to drive itself into the endometrium. Also, the syncytial trophoblast is involved in the production of human chorionic gonadotropin. This is released by the syncytial trophoblast to help sustain the production of progesterone by the corpus luteum. We already said that after the release of the egg, the shaft, which is referred to as the corpus luteum, will engage in the production of progesterone, which is also referred to as a pregnancy hormone. And also, this hormone is a biomarker in pregnancy test. When the serum or the urine level of this hormone is highlighted, it is used to stage pregnancy, for pregnancy to be confirmed. So there is the formation of lacuna, which are spaces created within the syncytial trophoblast. So this is one of the lacuna, and this is another one created within the syncytial trophoblast. And if you look at this structure, and as this develops or progresses, there's going to be a connection between one space and the other. These tend to create the lacuna network. So when there's a connection between one lacuna to the other, it's just to create a local circulation kind of, because at the end, the maternal blood that is seen in the endometrium lining, which are from the spiral artery, we supply the lacuna so that product from the maternal blood can reach the developing structure. So at the end, you have this kind of presentation. This is what we see at the initial state when these spaces are created. And of course, there's going to be a connection between one of the space and the other, and that will form the lacuna network. And after that, there's going to be the filling of this space by the maternal blood, which is from the spiral artery. And this will create the uteroplacental circulation. This circulation will provide at this moment nutrients and oxygen for survival of the developing structure. And this will occur around the 12th day of development. So this is the lacuna that is filled with the maternal blood, and this is the lacuna network that is created, connecting one space to the other. It is good for us to know that there is a fimbrium plug at the point where the blastocyst is implanted into the endometrium lining of the uterus. So there's going to be a form of closure at this point after the complete implantation of the developing structure. So this tends to create like a plug that helps to cover the opened region, and this is the fimbrium plug. There's also the studial formation. The studial formation is a modified elastic mucosal lining of the endometrial wall, because we know that the developing structure will be embedded within it. So there's a form of transformation of the mucosal lining into a form of elastic characteristics, so that as the embryo is developing, there's going to be space for expansion. Is divided into 3 subregions. We have the region of the endometrial lining that actually envelop the developing structure, and that is called the studia capsularis. Then we have the region that connects the developing structure with the wall of the uterus around the region where the placenta is formed, and this is called the decidual basalis. Then we have the last region that encloses the internal wall of the uterus, and that is called the decidual parietalis. And they are divided based on the regions where they are lining. So let's go to the transformation of the embryoblast. So let's look at how the embryoblast transform into the development of bi-laminar germ disc. So this is the embryoblast, and their cells are rearranged into an upper layer and a lower layer. The upper layer are tall epiblastic cells, or the primitive ectoderm. That means primitive at this stage. This will further develop into the ectoderm. and this is the epiblast. Then we have the hypoblast cell, which are cuboidal type of cell. They are located below the epiblast. And this was also referred to as the primitive endoderm. This means that they will further be transformed into the endoderm. And this is the formation of two layers. Remember when we started this lecture, we said that the second week of development is also referred to as the week of twos, which means that the structures that are being transformed into are always in twos. So the transformation of the embryoblast is also in twos. So we have the development of the epiblastic cell and also the hypoblastic cell. So this is the formation of the bi-laminar germ disc. And this is the cuboidal hypoblastic cell or the primitive endoderm. And there is the formation of the amniotic cavity, a cavity that is created within the epiblastic cell. This is the epiblastic cell above, when a small space is created within the epiblastic cell, and this is the amniotic cavity. And surrounding the amniotic cavity at the upper part is a differentiated type of cell that helps to form the boundary of this cavity, and this is called the amnion. This amniotic cavity will be filled with fluid and will be called the amniotic fluid. And this fluid tends to protect or surround the developing foetus so as to help to cushion it or protect it from impact. Further, from day 8 to day 9, we have the formation of the primary yolk sac. How is the primary yolk sac formed? We already talked about the embryoblast being differentiated into the epiblastic cell and the hypoblastic cell. From the hypoblastic cell, we have the proliferation of another type of cell that begin to differentiate from this region. And you see them lining the internal wall of the cytotrophoblast. And as soon as this differentiation is complete, this blastocele or the blastocyst cavity will now be transformed into the primary yolk sac. And this region is the primary yolk sac. This membrane that differentiates and it's seen to line the interior wall of the cytotrophoblast is called the exocoelomic membrane or the Elsas membrane. So we now see that the blastocyst cavity or the blastocele that was initially formed during the first week of development is now being transformed into the primary yolk sac. This primary yolk sac will further be transformed into the secondary yolk sac. This we would see during the course of this lecture. So this is the primary yolk sac, and this is the exocoelomic membrane or the Elsas membrane. So the embryo at this stage of development will now be seen with two cavities, the amniotic cavity and also the primary yolk sac. When we begin this lecture, we said that the second week of development is also referred to as the week of twos, the embryo having two cavities, which was initially one. So from day 10 to day 12, we have the formation of the extraembryonic mesoderm. The extraembryonic mesoderm will develop between the primary yolk sac and the syncytial trophoblast. The extraembryonic mesoderm, extra because it's outside the developing embryo. So it's a mesoderm that develops outside the developing embryo. So you see it developing between the primary yolk sac and the syncytial trophoblast. This, of course, has a reason, and we'll see that as we go through with this lecture. So this is the extraembryonic mesoderm highlighted in green. As development proceeds, there's going to be the development of cavities within this extraembryonic mesoderm. And these small cavities will come together, will coalesce to form just a single cavity. And that is where we now have the formation of the extraembryonic coelom or the extraembryonic chorionic cavity. So you see a cavity created within the extraembryonic mesoderm within this space, but the creation of the cavity is not in the entire region where we have the extraembryonic mesoderm. There's a specific region that is left blocked up, and in this space we have the creation of the connecting stalk. And the main essence of this is to create connection between the developing embryo and the fetal membrane. So this is where you have the connecting point, and this will tend to create a means through which the developing structure will be connected to the fetal membrane. So you can see that the extraembryonic mesoderm has the basis for its formation. And apart from creating a cavity, it's also able to create a connecting stalk that helps to connect the developing embryo with the fetal membrane. So the creation of the cavity does not occur within the entire length of the extraembryonic mesoderm, just for this reason. Because of the creation of cavity within the extraembryonic mesoderm, it now tends to divide the extraembryonic mesoderm into two layers. We have a layer that is connected to the primary yolk sac, and you have another outer layer that is connected to the syncytial trophoblast. So the layer that is connected to the primary yolk sac is called the splanchnic extraembryonic mesoderm, while the outer layer that is connected to the syncytial trophoblast on the outside is called the somatopleuric extraembryonic mesoderm. So you can see that the creation of cavity now divides the extraembryonic mesoderm into two layers, an inner layer and an outer layer. So let's look at the chorion. The chorion is a membrane that covers the fetus in the later stage of development, but it is good for us to highlight the structures that forms the chorion during the 2 weeks of development. The chorion is formed by the collection of 3 structures within the outer part of the embryo. So structurally, it is made up of the syncytial trophoblast, the cytotrophoblast, and also a specific region of the extraembryonic mesoderm. Remember, we talked about the extraembryonic mesoderm, which is the mesoderm that is created outside the developing embryo, but of course divided into 2 layers. We have this splanchnic extraembryonic mesoderm that remains connected with the primary yolk sac, while we have the somatopleuric extraembryonic mesoderm that connects with the syncytial trophoblast. So the specific region of the extraembryonic mesoderm that forms part of the chorion is the somatopleuric extraembryonic mesoderm. So these 3 structures form the chorion, and the chorion here in the later part of development that surrounds the fetus is highlighted in orange. You can see the fetus and you see the membrane covering it. So in going to the basics and trying to highlight these different structures that form the chorion during the 2nd week of development, we can say that it is formed by the syncytial trophoblast, the cytotrophoblast, and also the somatopleuric extraembryonic mesoderm. So these 3 structures will later become the chorion during the later part of development. This is the developing embryo on the inside, and you have the fetal membrane around it. You can see that it is structurally placed in the quest of forming the chorion later in the developmental process. So what we see in the 2nd week of development is already programmed to suit what is going to happen later in the developmental process. You can see how interesting this is. So further development is the transformation of the primary yolk sac into the secondary yolk sac. We already know that we have a large primary yolk sac that is formed. A larger portion of the primary yolk sac is chopped off, and this will then transform a large primary yolk sac into a smaller secondary yolk sac. So you have a smaller secondary yolk sac being created after the reduction of the primary yolk sac, and you have the remnant of the yolk sac, which is a region that is detached from the primary yolk sac. So the secondary yolk sac remain connected to the developing embryo. You can see that they still stick with it, while the remnant of the yolk sac is detached from the developing embryo. So this is the secondary yolk sac, and this is the remnant of the yolk sac. So at the end, what we see is the developing embryo at the center that is connected to the fetal membrane through the connecting stalk. So this is the connecting stalk, and this connecting stalk will later become the umbilical cord during the developmental process. This is the final presentation that we see in the 2nd week of development. That's the bi-laminar jump disc. This is the connecting stalk that connects the developing embryo to the fetal membrane that is formed by the syncytial trophoblast and the cytotrophoblast. Then you have the cytotrophoblast also proliferating and transforming into the primary villi. And of course, they drive into the syncytial trophoblast, where we have the establishment of the enteroplacental circulation. And this is so created so that the maternal product can reach the developing embryo for survival by providing it with oxygen and nutrients. We can check our understanding of this lecture through the following questions. The first one is, what specific structure produces the human chorionic gonadotropin? Also list the structures that form the chorion. And the last question is to outline the final presentation of the embryo on day 14. This we've highlighted in details during the course of this lecture. I'll be expecting your responses in the comment section so that we can drive more on this. So thanks for watching this video. Let's meet in our next lecture class.